All right, everyone. So hey, everyone. So this topic for me has been a long time coming. I've been wanting to talk about machine learning and biology, just because it's insane what's happened in the space. Like in the past year alone, like various like biotech companies are growing like 7x, 10x. We had like a COVID vaccine like developed in 2 days. Like we have things like DNA printers. We had AlphaFold. And so generally, this is, I think, a space where the impact is, borderline sci-fi, but it is stuff that people are actually building. And so a lot of these innovations are unlocked by machine learning, and people sort of overestimate how complex it is to get into it. That said, it's sort of like, easy to get into, difficult to master, as most interesting things are. But I thought, like, in this video, I wanted to give you sort of a review of like all of these like references, so you can get like a much better sense of like what this space is like. So, at a high level, I think one analogy that will really help you is that, like, there's sort of like a few things that we're going to be working with. So, we have the concept of DNA, right? And so you can think of DNA, well, it comes like in this like double helix or whatever. I don't know if I can draw a double helix. And I want you to think of this as similar to disk or storage on your machine. So this is basically your genetic code. But then to turn like you have this other thing called like RNA, and you can sort of think of this like your RAM. And what RNA does is it takes DNA. Yes, I'll do another picture. So like uh, if you have DNA, then RNA can create amino acids out of it, right? So, and you can think of amino acids as sort of building blocks. So yeah, I mean, let me do this. So, like, let's say this is storage, this is RAM, this is your syntax, and a whole bunch of amino acids together create like a protein. And you can think of a protein as a sort of like biological program. So I know this sounds like very abstract right now, but this analogy will really help you to understand what's going on. And so, let me just start walking through some of the references, and it will make this point very clear. So I'd say like my favorite introduction to proteins was this article by Khan Academy. It's not an article, so it's sort of like a short online class, and there's like really nice videos. But I just want to show you uh, the lecture notes. So what do I mean by biological program, right? So there's various like basically a protein has some sort of like biological function. It can be an enzyme, which means it can speed some reactions up. It can be a hormone, which means it can control some physiological processes like growth or development, reproduction. And there's like you see like a whole bunch of them. Like like their function can be transport, it can be hormone signaling, it can be defense, it can be contraction, it can be storage. So really, any sort of biological function is orchestrated by a protein. And one of the main things we're going to discuss, right, is that like a protein in of itself is a sequence of amino acids. So think of it like if you have like this, like let's say this is A, etc. And this is sort of like it as a sequence. But actually, the sequence has a sort of shape in 3D, so it could look more something like, instead of it being looking like this, well, actually, if in physical space, maybe it looks more like this, for example. So it has a certain structure, and this structure determines its biological function. And so then when you're asking yourself, like, well, why is AlphaFold a big deal? Because you can predict the structure, i.e., the function of a sequence. And the reason this is a big deal is because gene sequencing is cheap. There's like some machines that do it, and I'll show you what they are in a second. Whereas like predicting structure is expensive. So AlphaFold basically makes an expensive problem cheap. And this is why it's such a big technical innovation. And this has all sorts of applications, right? Like, so you noticed, like if you look at the applications here, this can range from like making like more digestible food to having like better transport for drugs for defense, so stuff like vaccines or storage. So like making crop like more resistant and better. Uh, so basically, like the range of applications is unbounded. And if you know if you're excited generally about computers and you think about like the kinds of stuff you can do with a computer, you can do a lot more with a biological computer. And so I think I personally find this to be the most exciting trend in technology today. And so I hope this will get you interested in how to program biology. So so I've been talking about these things called amino acids, right? And so there's 20 of them, and they all have like the same structure. Like they have these like alpha, like they have these alpha carbons, they have a side chain, and they have a carboxyl group. So uh, how this works doesn't really matter too much because it's almost like, well, you don't really need to know how circuits work to be able to program computers. Like it certainly helps, but the layers of abstraction make this really nice. And so if you abstract this away, you'll notice, like there's like here 20 or 21 of these. Like there's like aspartic acid, there's a glutamic acid, glycine, asparagine. Like there's a whole bunch of these guys, right? And so, but instead of thinking about these as like these sort of like molecules, well, you can think of them just as a letter, right? And so then then a protein will look like, let's say, RHKDE, for example. So a protein could just look like something like this, like RHKDE. Right, so this is a protein, and then these are each an amino acid. Right, so zooming back out, we know basically DNA is important because it's sort of like a storage mechanism. RNA helps you create amino acids, which are basically like a vocabulary or a syntax for programs, biological programs, which are proteins and have all sorts of functions. Right, so and when we talk about like the structure of proteins, there's sort of like a four kinds of structure that we typically think about, right? So far I already hinted at two forms of structure. So the first one is just the sequence, right? Like which amino acids come one after the other. So this is sort of like you can think of this as a vector. And then typically proteins, depending on how they're binded on how they're binded together, like the hydrogen, basically the hydrogen bondings make the amino acids fold into certain repeating patterns, and this could either be like an alpha helix, so like DNA has a double helix, or it could be like these pleated sheets. And when we think about here, like the tertiary structure is really what a lot of research papers think about, and what the tertiary structure is about is that it gives you the three-dimensional shape of a protein. And so when you think of like you'll sometimes hear the term like contact prediction. Like that means at which points does the protein fold onto itself? Because that'll just determine the function. And then quaternary, like I haven't seen too many papers talking about this. But this is like if you have like multiple amino acid chains, so it's like basically several proteins all intersecting into each other in a larger protein. But you can sort of think of like you can assume that this is fairly similar. And generally, I would say for the rest of what we're going to be talking about today, we're really going to be talking about the primary protein structure, and we're going to be talking about the tertiary protein structure. And so, like generally, if you find this interesting, like I mean, this was sort of like a speed review, right? But if you find this stuff interesting, there's a bunch of books that I would really strongly recommend you buy. So the first one is this one, like The Machinery of Life by Goodsell. So the basic of this book is almost like a piece of like a work of art. Like there's like these like beautiful figures, and it gives you like a very nice like visual sense of how all this stuff works. Just one warning, typically in biology, my main issue with biology books is that they tend to not cover things from an experimental standpoint. And what I mean by that is that they'll just dump information. They'll be like, well, this is how this thing works. And here's like, it's like very complicated Latin name that takes up like half a page, but they won't necessarily give you some good sense as to why it's the case or how did people discover it. That said, I think this book does a better job than most because it has like, these such beautiful, vivid pictures and very clear explanations, and it's very short too, by the way. It gives you a much better sense of how this stuff works. Another book that I really like that I just started reading is this one. So this is by James Watson. So basically one of the discoverers of DNA, like because it's Watson and Crick, right? who discovered the double helix structure of DNA. So I just bought this book, but I've heard great things about it, and it's also like very visual and very well written. And typically, again, reading something by the inventor, I would say is just generally probably more fun than reading it by secondhand, uh, by secondhand sources. So if you're interested in the history of DNA, how it was discovered, uh, I definitely strongly recommend this book. And recently, one of the things that sort of made me really want, even though I'd been looking at DNA and genomics and proteins for a while now, this was sort of one of the articles that I read over the break that made me really, really want to get into this again. So this is an article on by like so this guy calls himself like Powered DNS. I think his name is BERT. And basically, in this article, he sort of talks about how is the COVID vaccine developed, like the Moderna one. And so basically, I had no idea, but there's these things called DNA printers where you can give it the chain you want. And so, by the way, the difference between a DNA and a protein from a syntax perspective is that proteins basically have a vocabulary. So sorry, not proteins, amino acids. Have a vocab size of about 20, right? So this is what I showed here earlier. But DNA, on the other hand, has a vocab uh, of size 4. So this is just ACTG. And so if and again, like, and even though these are just like characters, like this is an abstraction for actual like molecules, right? Like it's like nature doesn't have ACTGs. This is just names for the physical stuff that we observe. But this abstraction is helpful because then we can sort of now treat DNA as code as opposed to treating it like just a bunch of like molecules. So turns out there's like these DNA printers, and it's kind of nuts that these things exist. But they give you some good sense of like a source code. So today it's actually very possible to just inject DNA into sequences. You do this by having like a start signal, like an end signal. And to sort of show you here, like when you think of like DNA in general, uh, here you can see here like okay. So first off, you'll notice here that you have uh, like first off you have like these three characters that are put together. So these three characters are called the codon, right? And so if you have like yeah, so here you go. So each three characters on RNA is a codon, and every codon encodes for a specific amino acid, right? And so this is how you get to go from a vocabulary of size 4 to a vocabulary of size. Think about it, right? So if you have three characters and you have and you have four choices for each one, then you have 4 by 4 by 4. So you have potentially 64 different codons. Yeah, so it's right here. So 64 different codons you can encode. But there's only 20 different amino acids, which means that a lot of them end up being duplicates. And so this is what the table ends up looking like. And so there's a lot of duplication in nature in general. So this is, I think, a theme covered in Talib's like anti-fragile book. But just to give you a sort of sense of it, is like, well, there's a reason in nature, well, why, for example, we have two kidneys, we have two lungs. And that's because like replication makes you more sensitive to failures. And so the joke there is like, well, if an economist were trying to design the human body, they would design it with one kidney. So hey, Krishna, nice to see you again. And so basically, this is sort of like how you can go from like characters in DNA to characters in proteins and generate stuff. And at this point, it's actually very easy to inject like basically to inject code in DNA and change it and print it. So yeah, we are discussing AlphaFold, but this is, I think, a more general introduction. But AlphaFold is going to be one of the things. The full set of stuff that we're going to discuss is all of this. Uh, so remember now, so let's just forget about AlphaFold for a second. Let's talk about a simpler problem, which is BERT on proteins or on genes, right? So typically we said, like, let's say we have this protein, right? Like RHKDE. So this is a sequence. So the question now is, can we turn this into a structure by using a neural network that works really well over structure? So basically a transformer model. right? So basically a transformer model, and then you get a structure. So how does this work exactly? And so what I liked about this approach is this was sort of like my introduction to this whole field, as in you don't need to understand what proteins or genes are to do this. You can just treat them as characters in a vocabulary and then just do this, right? So one example I wanted to show you, for example, is this was a recent Facebook research paper that does essentially BERT on proteins. And so you can see that they have like in their constants folder here, they just have like these all of these tokens. So these tokens, instead of being words or characters from the alphabet or characters from a dictionary or characters from a bunch of dictionaries, it's just like these 20 things. So if anything, doing language modeling on proteins is simpler than doing it on natural language because the vocabulary is much smaller. However, the structures you get are extremely complicated, and this is sort of like where this gets interesting. So the problem we're going to look at solving now is, given a sequence, right, can we predict the contact points using a transformer? So this is sort of like going to be how we're going to view this part of the problem. So as far as as far as I know, the first paper that sort of discussed this approach is this one. So this is some work by Salesforce Research and a bunch of other folks. So Jesse Vig, by the way, is one of the inventors of the Vig attention head visualization. So I have a separate stream just on that, so you should check it out. And so the way this works is that you're basically trying to figure out, let's say, given a sentence, you're trying to figure out for each word which other words did it use to be able to come up with its like own meaning vector, right? So for example, here like the, for example, should be like turtle, for example, should be paying attention to the, and then like let's say the rabbit quickly hopped, so hopped should go back to rabbit as it does here. And so this is sort of like a nice way to visualize these relationships. And so you may have guessed already what we're going to try and do here, but the main idea behind running BERT and visualizing attention heads on proteins will be to figure out which amino acids in a protein sequence pay attention to each other. And the ones that pay a lot of attention to each other are going to be the ones that have a contact between them. So let's just look at the paper and figure out how this works. Right. So like I said, so here like you'll see like there's like a really nice like list of co-authors like Eamon Cheong and Richard Tosh are very famous. I also think Jesse and Ali are very interesting to follow on Twitter, so you definitely should do that. And so here, so you can see, for example, like this is sort of like what a protein would look like. And then so sort of figuring out like where the contact or folds happen is sort of like where we're like what we're trying to do here. And for example, here, like they have like certain amino acids, and they can actually visualize, well, for a given amino acid, which other amino acid is it most paying attention to given like in each specific layer. So for example, here, for example, you can see that in this layer, there's like this very strong correlation. And then in this layer, there's a very strong correlation. So this gives you some sort of visual sense of how this works. And what's really interesting about it is that if you take the attention similarity of all amino acid pairs. So let's just take, for example, on average in the entire data set. Yeah, so Krishnan's asking, I thought Salesforce was some sort of a CRM company. I didn't know that they did this kind of research, right? So Salesforce Research has like world-class natural language processing researchers in general. And I think the reason they do is because they use their natural processing expertise to automatically do stuff in CRM. So for example, is this customer angry at me? Can I automatically extract entities? Can I figure out at which part of the life cycle a contract is? And so the reason why they ended up doing this, I believe, is because their NLP expertise ended up being very relevant at working with proteins. And I wouldn't say like that they're specifically in this paper, it's not like the protein work is world class. Like in the AlphaFold paper, I would definitely argue that it's world class in terms of what they're doing with proteins. But the benefit of this is this was sort of my entry point to this work, as in, well, I know some stuff about transformers. Now I can do protein stuff. So here, for example, like darker means higher attention, And then we're checking the pairwise similarity of all amino acids to all amino acids to each other. And this ends up looking a lot like this substitution matrix called the BLOSUM, which is a handcrafted, think of it like a handcrafted attention matrix that people traditionally use to predict protein folds. Right? And so let's see here. I think there's a paper that they don't show here. Yeah, I think, let me show it to you. ProcGen. So Brian McCann. Yeah, so Brian McCann recently left Facebook. He's working on search right now. But yeah, this is the paper I'm talking about that I also thought was very interesting. So let me just keep a reference to it here so that I'm making it a point now so that my viewers can actually look at my references easily. Language modeling for proteins. Hope this makes things easier for people. But yeah, anyway, like they had a really nice picture I want to show you that I think expresses some of the points I've been making. So if you go here, the amount of protein structures that we have available in the, like basically as part of data sets hasn't increased all that much with time. In fact, it's like pretty flat. Whereas like protein sequences has increased exponentially because protein sequencing has gotten a lot easier. So here what they do in this work, for example, it's like an extension of like this pure transformer model where you're looking at attention head similarity. And like what they do is they also feed alongside the sequence some desired arguments. And then given these desired arguments, basically you're saying by desired arguments, I really mean function. You're saying, I want this protein to do the following. And then can you infer a structure that does this, right? Because remember, structure is equivalent to function in protein space. And so this is sort of like really interesting. And so this work was actually an extension of some work they did in traditional NLP, where the idea is, for example, you could say, well, say this words like in the words of Elvis, or say this in this style, say this in an angry way, or say this in a happy way. So this is sort of like an extension to this. And again, this is a labeled data set, like because there are protein data sets where the arguments are labeled. So again, this is a supervised learning problem. So as part of your loss function, you would have like, again, like when you think of, because this sounds almost too simple, right? Like let's say, this is RHDKE, for example, like this is your protein sequence. Like the supervised learning signal is you mask this and basically predict D given surrounding context, right? So transformer models are essentially a way to turn a problem that's unsupervised into a supervised learning problem. So then that's basically one part of your loss function, right? So this is, let's call this L1. And then L2 would basically be some arguments from a separate data set. Right? So moving on. Yeah. So there's not too much more here. I think that happens, but basically, I think you can sort of go through this if you like. I would imagine that longer sequences are probably harder. So what's orange here? Orange is accuracy. And then it's perplexity. Oh, actually, it's saying like for longer sequences are. Oh, no. Okay, so this is saying near the end parts of a protein, the model gets less and less perplexed, so it gets more confident of what it's actually seeing. So again, this is not like in NLP, for example. It's like sort of like it's like your unit is your whole paragraph, right? And so again, here as a number of conditioning tags also increases, the accuracy increases, which is very promising, right? Because it means that you're more likely to generate what you actually want. So let's just take a look at this like one last time here. Where was the paper? Let's see, anything else I think I can show you here that's interesting. Yeah, so here, for example, they look at the probability that where the attention head was mostly focused is in fact a binding site. Like again, like here it's like, well, not all sequences have a structure, but the ones that do have a structure, well, now you have a supervised learning signal and you can like learn that. So this is sort of like how they come up with this. And I actually thought this paper is like a very interesting proof of concept for how this stuff can work. Uh, and I think now, like, let's say we've been treating so far proteins as these abstract sequences. But now, like, let's start looking at geometry, right? So let's start looking at algorithms like AlphaFold and how they work. So actually, before we talk about AlphaFold, I wanna show you some easier examples, right? So this was, for example, a paper uh, by Michael Bronstein and a couple of other folks on basically what they call fast end-to-end learning on protein surfaces. So yesterday I had a separate stream about point clouds, and this wasn't a coincidence actually, because you can think of like a protein in a few ways, right? So you can think of it as a sequence, but if you look at it from the tertiary structure perspective, well, it's a 3D shape. And a 3D shape, you can also now think of it as a point cloud. Like imagine you're taking like a LiDAR for proteins, but LiDAR wouldn't work for it. It's a small, like, so it'd be X-ray crystallography, for example. And you're gonna get like what it looks like in space, right? And so now the problem is, can you do basically deep learning on these surfaces and point clouds? And the short of it is yes, like basically here, the image that makes it very clear is here, where you have like point clouds where the distance function isn't purely Euclidean. It's basically based off of geodesic distance. Basically, you navigating, like imagine that you were like a tiny particle, like walking along the sides and twists of a protein. So this gives you like a better measure of distance in this space because like Euclidean space isn't that meaningful in this space. And using these distance measures, can you then predict contacts? And again, this is a supervised learning problem, right? So you're instead of treating a protein as a sequence, you're treating it as a point cloud, but you still have a supervised learning signal that tells you where the binding sites are. Either these are manually placed or like some other machine learning algorithm has them. And then it all uses like what this data set called the protein data bank. So again, gives you like a very similar idea. And it's just like here, the point I want to really try and make is that depending on the way you view your problem, like you can view the same protein problem in 4 different ways. And depending on how you view it, they're all supervised learning problems because you either have contacts, and when you don't have contacts, you can generate them. Right, and so now let's start talking about AlphaFold for a second. So with AlphaFold, there's this nice like tweet level summary of it. I'm not going to cover it because I actually I actually thought the best reference to understand what AlphaFold does was by Mohammed Al-Quraishi. So let's go over his summary. So it's unfortunate, like I can't actually show you the paper because as far as I know, they only had like more marketing blog posts. But Mohammed Quraishi is like sort of like a domain expert in this stuff. He has many papers about protein folding. And so it's going to give you a pretty good sense of why this problem is interesting and sort of like the main key innovations. The main thing I want you to think about though first is here. So this is a paper of, so sorry, this is a chart showing basically, think of it like the accuracies of AlphaFold versus the next best methods. So basically AlphaFold was a big deal because people weren't expecting these kinds of accuracies. Basically here, like let's say they say, well, like above 95% accuracy in predicting fold is almost similar to predicting it experimentally. So there's multiple techniques, but the one I'm mostly aware of is X-ray crystallography, which you can think of it like taking like a picture of something frozen in space at a molecular level, and it works pretty much for everything. There's like lots of like nice videos about it on YouTube that I'd highly recommend you check out. But one of the main things I thought was interesting was, well, for a lot of like for some proteins, the difference in performance was relatively modest, right? So it's like, well, these couple of points, like for the really hard ones. But at some points, like there's like a huge difference. Like it's twice as good. So even like let's say the accuracy difference as an aggregate is about, let's say, what, 10-20% or something. Uh, for some points, you have like 40% differences, and so this is a big sign that like maybe there is a sort of like a breakthrough happening here, right? So what is really happening? So when people talk about protein folding, there's a couple of problems that people generally refer to. So there's the idea of how do you predict the structure of a single protein from a sequence. So this is what I've mostly been talking about today. Uh, but there's also like, let's say you can have like multiple proteins, or it could be the dynamic folding pathway. So like the protein moving over time, for example. Um, And generally, like these are all complicated, but we, like the thing is about like if you solve one, it becomes a lot easier to solve the others. And that's why it's not that like protein people are going to be out of a job, but it's that protein folding is probably no longer a scientific problem. It's now an engineering problem. So now it's about how do you make it better. And again, like engineering problems are hard because getting something from 99 to 99.9 is very, very difficult. You need to build a lot of infrastructure for that, a lot of data. But it tells you that it is solvable in principle if you're willing to throw a whole bunch of money at it. One thing I want you to note about AlphaFold is that the team isn't necessarily purely machine learning experts. Like they actually have some of the best like protein people in the world on the team. And I guess like otherwise it would have been very difficult for them to do this. So there's a few I think important ideas. There's a few important ideas here. So for example, here for example is they sort of like learn the MSA, right? So let's see here, because I don't remember this article by heart. Because are not blue share mount. Okay, yeah. So let me just show you what this is. So multiple sequence alignments. So if you look at like biological, no, that's not what I wanted. I wanted the Wikipedia article, yeah. So here, for example, so this is like a like protein, like various proteins from various families, and you're trying to figure out which ones are similar to each other. And so typically this is done like manually. We think it with like string matching algorithms. But I believe like these are exponential in the amount of time that they take to compute. And so the idea here is that AlphaFold has the option to choose which other sequences it needs to pay attention to figure out how it needs to fold, right? And so this idea that it can sort of do this recursively and sort of slowly build out a structure was one like difference. The other, I think, probably more obvious one is that it's like end-to-end differentiable in the sense that it doesn't have hand-coded parts as part of the its routine. So it's like the whole thing is so yeah. So sorry. So I was reading a question. So what is your general take of scientific problems being converted or perceived as engineering problems? So do you think eventually this would make domain experts redundant? I think what it really means is that it doesn't make domain experts redundant. It just means that domain experts need to learn a bit of software. So they need to learn some machine learning and some software to be relevant. But sort of treating it from a pure domain expertise, I think, is not likely to be fruitful. And I would actually say, I would actually argue it the other way around. I would argue maybe eventually machine learning practitioners are going to become redundant. And that's because the core skill set is no longer that complicated. Like the fact that, libraries like TensorFlow and PyTorch make it very trivial to use language models, to use like ResNet, to use GNNs. Like this is not that hard anymore. And so I think what's going to happen is that if you have some DevOps skills and basic software skills, you can learn the basics of applied machine learning. And then the way you would make your career interesting is you would have some domain expertise, like let's say proteins, like econ, physics, and then there you're really at a point where you can do interesting stuff. The issue is like traditionally in machine learning, we sort of treat as part of our domain expertise like language and images. So those are two areas where it's become sort of like our comfort zone. Thank you for the new subscribe. I really appreciate it. And so, but Sloopez, cool name. But yeah, so I generally think that this is why sort of going very deep and learning all the machine learning techniques, I think is not likely to be as productive as it was 10 years ago for some people and earlier in their careers. So I think having some domain expertise will be invaluable. So just follow your curiosity, like whatever you think is interesting, you should follow. So another touch that I thought was really interesting was this idea of equivariant transformer network. So I had a tweet talking about this, but let's talk about this for a second, right? So when you have like a convolutional neural network generally, like a CNN, right? You have like, let's say this is the larger image, it's highly abstracted. And then you have a filter that you're passing along, uh, that you're passing along here. And so this gives you basically some more, like this gives you some like nice properties like translation invariance, right? But a really popular data processing technique, oh, thank you, Stupas, I appreciate it. So, like a really popular data processing technique for CNNs is what's called like domain randomization. So, let's think of this thing. So, let's say you have like an image, let's say of a, I don't know, like a human. What you could do, for example, is you can scale it. You can make it grayscale. You can crop it. You can rotate it, etc. Like you can do also like you can basically saturate it. You could make it all gray, like all pink. That's what saturated means. So there's all sorts of data processing pre-processing techniques you can use that essentially fake how much data you're giving to your CNN. So as opposed to, let's say, having like 1,000 images that you feed to CNN, well, you could make this 1 billion that you feed to your CNN, and ideally this would make it generalize a lot better. And so here specifically with CNNs, like they do very well with translation invariance, but they don't necessarily have a good solution for rotation invariance. And so this is what SE3 networks are. So what's SE3? So I have like, this is more in my book, The Robot Overlord Manual. I talk about SE3, but SE3 is called like the special Euclidean group. And let me just show you a picture here. If I go to Google Images, that's definitely not what I care about. All right, so I know this image is kind of complicated to look at, but what I sort of want you to think about here is that, well, what's a group, right? Like, I did not expect us to be talking about group theory, but it's okay. So basically, group theory is the study of symmetry, right? So for example, uh, so groups theory is the study of symmetry. So for example, like let's take you have a square like this, right? How can you rotate it? So let's say you rotate it by 90 degrees. You're gonna end up with something that looks like this. If you rotate it by another 90 degrees, so like 180, it's also gonna look like this. But if you rotate it by 45 degrees, well, it's gonna look like this, right? And so you're closed under symmetry for 90-degree rotations and multiples, but not for 45 degrees. Like you end up with a different element. So the general, the general mathematics that you would use to study this is called group theory. So the reason this comes up in the context of proteins is because, like, well, let's say you have a protein that looks like this. Well, that's the same as a, let's see if I can flip it. Yeah, so that's the same as a protein that looks like this, right? So this is equal to like this, right? But because we're dealing with the Euclidean group, well, this is also equal to this. And this is also equal to this. So these are all the same protein under SE3. And because the reason why this becomes important is because it sort of lets you compress the complexity of your model. So like your model actually lies on a lower dimensional manifold. Like I talked about this yesterday, talking about point clouds. But the fact that these are all the same means that you have like less things that you need to discriminate between. And so you can basically add modules that instead of having a CNN would be an SE3 CNN. I will let you refer to the original paper. Maybe I'll discuss it. I had a tweet about the paper. There was an accessible blog post about it. But yeah, and so that's pretty much it. So actually, no, that's not it. Sorry. Like, let's go to the Alphafold paper here. So I just want to end this on a note. So why is this all a big deal, right? So the reason why like making any sort of breakthrough in biology is a big deal is because one is you can synthesize proteins. So synthesizing proteins is a big deal because it means you can synthesize drugs, vaccines, crops, you know. So like we're going from like, okay, like here's a cat classifier to here's a cure for, like here's a cure for disease and how to reverse aging and how you can be immortal. And so obviously we're not there yet, but the fact that these problems are now like in reach that we can think about them, for me is the single most exciting thing going on in technology today. Maybe alongside Bitcoin, even though Bitcoin is very simple and that's what makes it exciting. But I generally think that this idea, like oh yeah, so there's like variants, like let's say variant prediction, uh, synthetic biology. So these are all variants of the protein folding problem, or like basically modules that build on top of the protein folding problem. But once that's solved, like again, more of these become engineering problems, and why this becomes exciting. And so remember, like really, the main thing is that we have a lot of sequences like this. We have a lot of sequences. These we don't have so much structures. We don't have so much. So the easier we can get to doing this, now we can sort of start synthesizing proteins with specific functions. And so I would highly encourage you, if you're interested in exploring this space, like sort of like an interesting project would be, you could just go to the protein data bank, right? So you can just go to the protein data bank and then you download some data from here. And then you can just go to Hugging Face and you can download a transformer and see what kind of good perf you can get with language modeling. And then once you're done with that, now start adding the geometric pieces from like looking at geodesic distances, from looking at GNNs, to looking at equivariant transformers. So basically the sky's the limit, but this is sort of like the core foundation that you need. Like you have a transformer and then you have stuff that builds on top of it. So yeah, I know this was a lot. Like we covered like a shit ton of references, but I hope this was interesting to you. And remember, if you get confused, remember this analogy is very helpful when thinking about protein biology as a computational problem. Like DNA is storage, RNA is RAM, amino acids are like vocabulary or syntax, and then proteins are programs. And programs can do a whole lot in the biological world, a lot more than they can do in the physical world. So yeah, I hope this was interesting. If you enjoyed this, please make sure to subscribe. And if you have any suggestions for future videos, please let me know. And thank you.